Today we will begin part 2 of Cell, the Unit of Life, lesson number 5. What are the main parts of a cell? Cell wall, cell membrane, nuclear membrane, nucleoplasm, nucleoli, chromatin fibers. So cell wall is the outer boundary of a cell and it is found only in a plant cell. It is absent in the case of animal cell. Cell membrane, it is plasma membrane. It is the second layer after cell wall and it is the first layer in the case of an animal cell. Inside the nucleus, what are the main components present? Nuclear membrane, nucleoplasm, nucleoli and chromatin fibers. Inside the cytoplasm, what are the parts present? We have endoplasmic reticulum or ER, mitochondria, the powerhouse of the cell, Golgi apparatus or Golgi bodies, ribosomes, the site for protein synthesis, lysosomes, centrosome, which is found only in animal cell, plastids, which contains the pigment chlorophyll to produce food by a plant, granules, vacuoles, and fat droplets. This is the structure of animal cell. Each part has its own functions. So we can see there is no cell wall present here and other components which is present in animal cell are performing its own functions that we'll be discussing later. This is the structure of a plant cell. You can see the violet color or the purple color thing is the cell wall and green color thing is the plasma membrane. And we have rough endoplasmic reticulum and smooth endoplasmic reticulum. ER means endoplasmic reticulum. Mitochondrion, nucleolus, nucleus, and we can see a specific feature compared to animal cell, that is large vacuole is present in a plant cell. These are the differences between plant cell and animal cell. There is a definite cell wall for a plant cell, but it is absent in the case of animal cell. Plastids are present in plant cell, it is absent in animal cell. Centrosome is present only in animal cell and large vacuoles in plant cell and small vacuoles in animal cell. And cytoplasm is very densely packed and granular. Densely packed means thick in the case of animal cell. And the reserve food material is in the form of starch. So glucose is produced by photosynthesis and changed into the form of starch and stored inside the different parts of the plant. It is in the form of glycogen in the case of animal cell. Compared to animal cell, the plant cell is bigger in size and having a rectangular shape. These are the differences between prokaryotic and eukaryotic cells. First, you should know what are prokaryotes. Here, the prokaryotes are having specific structure. Examples, bacteria, blue-green algae. Blue-green algae means BGA or cyanobacteria, which can fix atmospheric nitrogen directly and supply to the plant. Then we have euglena, amoeba, all the plants and animals which are coming under eukaryotic cell. So compared to prokaryotes, eukaryotic cell is bigger in size. It is 5 to 100 micrometer. So how much is 1 micrometer? It is given in your textbook, 10 raised to minus 6 meter. So that is a size difference between prokaryotic cell and eukaryotic cell. And there is no specific nuclear region in the case of prokaryotic cell, but there is a well-defined nuclear region in the case of a eukaryotic cell. And there is a single molecule of DNA as chromosome, the case of prokaryotic cell, but there are many the case of eukaryotic cell. Nucleolus and membrane-bound cell organelles are absent in the case of prokaryotes. It is present in the case of eukaryotes. So this is about the differences between prokaryotic and eukaryotic cells. What is a cell wall? I told you the first layer which is making the plant cell is cell wall. It is helping the plant cell to protect from unfavorable conditions like temperature or any other unfavorable conditions. Here, this is the perfect component or feature giving rigidity and shape to the plant cell, and it is made up of cellulose, a complex form of carbohydrate, or we can call it as polysaccharide. Here by osmosis, osmosis means higher to lower concentration movement. It is making the plant cell shrinking by a process called plasmolysis. Next is the main or the important structure of plant or animal cell. It's called as plasma membrane or cell membrane. This is deciding whether it should allow any outside particle to get entry inside or any inside particle to go outside. So like security guard, it is doing its duty. Even it is protecting the cell from injury and giving an outer boundary to the cell. It is a type of feature present in plants and animal cell helping for active transport of carrier proteins. Sometimes to engulf the food, engulf means to capturing the food, it is using its plasma membrane part by doing some modification, thereby it is engulfing its food by a process called endocytosis. Here, the differences between diffusion and osmosis we are going to see now. How does the movement of substances take place in and out of cells? By two processes called as diffusion and osmosis. Both are similar, but there are some differences between that. You can see the definition. There is movement of molecules from higher concentration to a lower concentration. It can be any kind of matter. Here it is not specifying which kind of matter. So any kind of matter which moves from higher concentration to a lower concentration, this is named as diffusion. So example, your inhalation or exhalation, you have more oxygen outside, so higher concentration of oxygen outside, and inside you is less in concentration. So higher to low means oxygen is moving from outside to inside. So this oxygen is moving from inside to outside for this. That is example for diffusion. Next is osmosis. This is also given higher concentration to lower concentration. So what is higher concentration to lower concentration? Here you should know higher concentration means higher concentration of water molecules, higher concentration of solvent to lower concentration of solvent. So don't get confused here. The number of water molecules is giving higher concentration. So where more water molecules are present, that is giving higher concentration. From there, the movement happens to lower concentration. The importance of osmosis. Here, the importance of osmosis. Example, the entry of soil water into root. Because here, water concentration is more outside the root. So it is moving from higher concentration of water to lower concentration. So in this case, the stomata opening and close, that is also related with increased or decreased osmotic pressure of the guard cell. This is also related with osmosis. Next is, how can you prove this osmosis? You need an egg for this. You're removing the shell of the egg or deshelling it by keeping in dilute hydrochloric acid for some time, so that the eggshell, which is a component made up of calcium carbonate, will be dissolved in that and gets removed. So now you have the egg ready for doing this experiment only with the thin outer skin. Put the egg in water for 5 minutes and observe. So how are you going to do this experiment? You're taking water, to this, you're adding egg. So you have an egg now. This is de-shelled egg. Okay, this is de-shelled egg. There is no shell for this. So now, what is the observation? You're keeping 5 minutes. The egg is swelling. What is the meaning of that? Egg is swelling because water passes into it by osmosis. So when you are getting weight more, you are eating more food. That is coming, something is entering into your body. Right here, the size of the egg is increasing because the water has entered inside from out to in. So here it is outside to inside entry is happened. That means higher concentration of water molecule is outside, lower concentration of water molecule is inside. Yes. So next one, we are trying the experiment another way by taking another beaker with water. It is mixed with more salt. Then keeping your egg, de-shelled egg here. So what happens? Here, salt concentration is more outside. If salt concentration is more, means water molecule is less outside, low concentration of water outside, and inside more concentration of water. So that entry happens always. Here high concentration of water is inside. So inside to outside, because salt is more outside. We are not considering salt concentration as more concentration. Always remember water concentration. So high to low, inside to outside. In this experiment, inside to out, inside to outside. So what happens inside to outside? Shrinkage of egg. These are the differences between diffusion and osmosis. What are the differences? The same thing, definition you can write. Then it occurs in any kind of matter in the case of diffusion, but osmosis only liquid medium. That's the main difference. Here it does not want any helper to do this process. There is no semi-permeable membrane. Here in the case of osmosis, it requires a semi-permeable membrane. And diffusion, as it is happening in gas, it is a very speedy process, but here it is water, it is a slow process. Here there will be equalization of concentration of diffusion. What is equalization means? Once the movement starts from higher concentration to lower concentration, when both outside and inside the concentration is equal, that time the diffusion stops. But that is not happening in the case of osmosis. Here, these are the three types of solutions based on this osmosis: isotonic, hypertonic, and hypotonic. Iso means same. Iso means same concentration inside and outside. Hyper. So hyper means here just three. Here concentration of water molecule in outside medium is lower than inside the cell. That is, solution is very concentrated. More water leaves the cell than entering it. That means inside to outside, into out. So here high to low concentration is moving. So the cell is shrinking. Hypotonic means out to in, outside to in. That means outside concentration is more, higher to lower concentration. So the cell will be swelling up, outside to inside. Inside the water is entering more. So this is about, these are the three types of solutions: isotonic, hypertonic, hypotonic. You should be able to explain it with the help of examples. So we have finished part 2. We'll continue with the part 3 later. Thank you.